Yeah, it's a great honor to invite Dr. Tsui-Fen Chou come here, join us. Tsui-Fen Chou's scientific journey began in Taiwan, where she earned her bachelor's degree in pharmacy from National Taiwan University in 1999, and later she moved from the tropics of Taipei to the icy lakes of the Midwest to pursue a PhD in medicinal chemistry at the University of Minnesota, under the mentorship of Professor Rick Wagner. And she completed her doctorate at 2006, focusing on the enzymology of phosphoramidate, hydrolase, and its implications for the rational design of the antiviral and the anticancer prodrugs. So later in 2007, Dr. Chou joined the Caltech as a postdoc fellow in the laboratory of Professor Ray Deshaies. Okay, there she pioneered identification of chemistry inhibitors of the AAA ATPase P97, applying structure-based drug design and high-throughput compound library screening. Now, currently, Dr. Chou is a research professor of biology and a biological engineer at Caltech. Her research program spans multiple areas of chemistry, chemical biology, proteomics, and translational science. She investigates physiological substrates of enzymes of unknown function, performs enzymatic and kinetic analysis to uncover critical amino acid residues, and develops small molecular inhibitors for targeted cancer therapies. With extensive expertise in Orbitrap, LC-MS, and MS-based proteomics, she has identified functionally important protein complexes and advanced new workflows for quantitative analysis. Expanding into neurodegeneration, Dr. Chou now applies induced pluripotent stem cells, iPSCs, derived neuronal models and brain organoids to identify disease biomarkers using proteomics. These models also provide a platform to evaluate therapeutic compounds for their ability to rescue phenotypes associated with the pathogenic P97 mutation in motor neurons and organoids. And welcome Dr. Chou to give the wonderful presentation.
Thank you, Xianghong. Yeah. And first of all, thank you all for coming and for the invitation. I had wonderful time talking to Xianghong and also the postdoc, just 2 hours before the seminar. And I think, you can ask me any question, interrupt me during the seminar. So, it's formal and informal. I can take any kind of whatever you like. And I actually been here 2024 for the Asian retreat. And also like the same year we visited before. So thanks for having me back. Nice place, and looking forward to future collaboration. And today I'm going to talk to you a project, and I will start with typically how do we use proteomics as a tool, mass-spec-based proteomics as a tool to find what's wrong with certain disease. And if we are lucky, we can understand what's wrong with the mutation-caused disease. We can design screen to find potential therapeutic compounds or a protein, even can be a drug to treat a disease. Oh, good. So since Xianghong already gave a nice introduction, this is just a timeline of showing you how I get to where I am now. And along the way, I learned as a chemist, learn how to do biology and learn how to do mass spec. And I feel also high throughput screening. All of this learning process actually helped me to address the question that we want to answer that in different approach. So, And then I always run out of time. So I want to thank people in the lab first. And you can see the people I colored red, Yanping, Diana, and Kent, and Will. Those are the people who that contribute to the work I'm going to tell you. There are other projects in the lab that I might not focus today, like P97. It was, we studied for a long time, and P97 associated disease or cofactor involved in how P97 regulates cell function. Today, I will give you like all of these now published data to give you a story how these new projects develop. And when I want to start a kind of advertisement and also tell you how we generally use mass spectrometry-based proteomics, and also the two gentlemen here are very important. They help me run this proteomic center at Caltech, and we work with people inside Caltech, also outside Caltech, even with company. They're willing to just pay complete full service to analyze, say, the drug conjugate, how the drug conjugate moves to the brain. We can tailor what people need for their research. And this year, and I have 3 instruments in the facility now. The first one is older, it's Eclipse, it's a tribal arbitrary instrument. Now we use it more for top-down or ascorbic labeling or measuring DNA, glycan, or small molecule metabolite. Because this year we just got a new, last year, 2 new instruments, one called arbitrary Astro. And this one now is the state of art. You can do single cell proteomics, you can do complex sample, and the last one is called Stella. And what we did is, most of the time we start with Astro. You can declare 8,000, 10,000 protein change due to the manipulation. You can knock down a gene, knock down a protein, or treat with a small molecule, or compare human sample from different time points for natural history study, and you find this that 50 proteins are differentially expressed. In all days, we have to go purchase antibody to prove what we found from the untargeted proteomics that really translate can be validated. Now we have the other instrument called Stella. What I envision to do, we are doing it, but not everybody is using it yet, is once you have list of protein, we can run the same sample. We don't need to re-prepare the sample on the Stella, but if only select the 100 protein with, say, like 3 or 4 peptides per protein, then you can validate. We don't need to waste money to find antibody to show, is this antibody working or that. So you basically can narrow down the false positive from the untargeted approach. So I'm quite excited to, last year we can get these 2 instruments. And the other thing I want to advertise is, so we work with NCI for cancer drug development. So NCI has this program called Next Therapeutics. What they want to do is they want to help academic people who may not have the expertise for certain, like high-throughput screening. If you apply for their program, they'll provide funding, provide expertise to help you screen for drugs. So about 2020, PAL, so the Proteomic Center I'm in charge, we are one of the functional drug discovery center. So what we did a lot is, they have this program and they have this drug, they want to compare of the other, they have already FDA approved drug. What are the biomarker change? What are the protein expression? If it's a kinase, what is the phosphorylation pattern change? So all of these, we can help, we work with them to help other people's project. So it's a good practice for people in the center to learn how drug discovery works. So if anybody interested in that, I can also tell you what they are looking for, how do you apply for a next drug discovery program. Now, this is the one I first described. We can from targeted approach to, from unbiased approach to identify a list of protein change, and you validate by Stella, which is the targeted approach. So this is published by the famous proteomic person, Matthias Mann's lab. So this is the workflow we are establishing now for the Caltech Center. And what type of proteomics you can do with mass spec? So this is a list of things from simple. We can look at protein expression. We can look at proteome mining, meaning there are certain species. We only have their, say, genome. We only have their RNA-seq data. We don't know what are the protein actually expression. We can do proteome mining. We can do functional proteomics. We can do structural proteomics. We can do protein-protein interaction and post-translational modification. So as we know, DNA make RNA, RNA make protein, but they are different flavor of protein. One simple PTM can make one gene to have 10,000, hundreds of different gene products. So proteomics is behind genomics, but with the advance of the instrument, I think we are improving every time when there's a new instrument came out. And this is the simple proteomic workflow. So you can come with any sample. Here I show you a cell plate, right? And this, for single cell, we normally need to isolate the cell and put it in a 384-well plate format. And you can analyze the protein expression at single cell level this stage. So with all of the improvement, with the best instrument, we can get 3,000 to 5,000, depends on the size of the cell, right? For immune cell, it's smaller, we'll get 3,000. For a bigger cell like mouse embryo, we can get more because it's a bigger cell. So we are excited with the current instrument we can do this. And one of my students, I will not tell you detail, but it's blackly, but she already have a bioarchive paper to do so-called spatial single-cell proteomics. So what it means is this is show you a picture of a kidney and versus healthy lung, and we can cut the cell based on what the morphology or based on what is the disease marker label, and you do proteomics on healthy versus disease type and see what is different from the same person or patient. Right, and this to show she has done for brain, for Parkinson's disease, and for breast cancer, and TB. Now, we just had this paper published in collaboration with another professor at Caltech. This is also a single cell platform. And this is a mouse embryo cell. The good thing is bigger. So you actually can identify more protein from the bigger cell. So the cell, I say, like the immune cell really is like 10 times smaller. So the protein you can identify just a lot less. And that's the limitation for the single cell still, but at least we can already detect 5,000. When I was a student, you can give a lot of sample, the mass spec only can detect 10 proteins. So the technology really developed a lot the past 20 years. And for this study, essentially, the lab, they can pull single cell from different stage of the embryo development. The one cell and per cell divide 2 and 4 and compare the protein expression profile. And there's even one experiment, the sperm entry point actually can also break the symmetry of when it's 2 cells. There are 2 cell types and how they can break. And again, this study is a lot of observational because it's really hard to go back to manipulate, to really do magnetic study. But we use this to identify the potential change. Then later you want to use other approach to study the mechanism. Okay, so those are all the background of what the proteomics center we can do. Now I will focus on the title. The title has the term called metamorphic protein. So I need to introduce you what does metamorphic protein mean. Metamorphic protein also means shape-shifting protein. So in the conventional wisdom is you have one DNA sequence and it will fold one protein structure or conformation, right? And you guys must heard about IDP or IDR, intrinsic disorder protein. What do those proteins mean? Means they don't have a fixed conformation until they interact with other proteins. But in those areas we call dark proteome because we don't have structure of this protein. Metamorphic protein is one type of dark proteome. It does have the structure, but its structure can switch from one conformation to the other dramatically, from alpha helix to beta sheet. And this, I just want to show you the conventional is one, actually, that should do this. Conventional is one protein and one structure, their function. And metamorphic protein, they have ability to shift the conformation to different state. And in one state, it has one biological function versus the other state, it has a different biological function. And here, just show you example of a few proteins. They are known to be a metamorphic protein. And the trigger for why the protein has to shift the conformation is different. Some of them is come from interacting with a partner. Some of them come from the environment. The oxidation of that protein will switch its conformation. So it's really hard to predict which protein can be a metamorphic protein until you really know that exists and there's the environmental trigger. So far, there are 96 proteins was predicted, identified to be potentially metamorphic. Again, these are because they are hard to estimate. And these are estimates. So we are lucky just recently received this foundation money from Caltech Foundation. We want to establish a proteomic method to search protein-wide scale. What are the protein can potentially to be metamorphic protein? And then we need to validate, right? And the reason we can use mass spec to identify was my collaborator, Andy and John, they had this concept that metamorphic protein undergo this cold denaturing property. So you can detect the protein conformation change when it's at cold temperature and it's partially denatured. So people might heard about limited proteolytic experiment. It has been used for over a century to determine protein conformation. You basically see what the regional protein is more flexible by protease digestion. And the past 10 years, people have linked the limited proteolysis with mass spec together to look at protein-wide how the protein conformation change can be addressed or assessed by proteolysis cleavage. So we basically apply that approach. The only difference by which with the staff scientists do this experiment is you do the protease digestion at cold temperature versus the hot temperature, and you compare for metamorphic protein, its cleavage pattern will be changing compared to cold and hot temperature. For the protein that is not metamorphic, monomorphic, which is green, it will not change. So like I explained to you, when we do the proteomic workflow, we have the protein, we count with the trypsin. We need to optimize the procedure because the temperature is different, right? You analyze by mass spec, and you can see the spectrum on this side are what you see on the mass spec detector. So you can see the green, which is the protein will not change conformation at different temperature. So its intensity will be very similar. For the blue protein, its temperature, conformation will change at lower temperature, so its intensity was different. So we already actually put this protocol in the bioarchive to only do E. coli in the beginning, and we apply this to the human proteome. So about half of the E. coli proteome we can detect because this you have to keep the protein in non-denatured condition. It's inactive, so we only extract protein by freeze salt with PDS. So about half of the total E. coli protein, like 2,500, we can detect. And among that, there are like 8% of E. coli proteome is metamorphic. So how does bioanalyze this? So as I mentioned, there's a mass spec protease cleavage at different temperature. And this is, again, the abundance of that particular peptide. As you can see, if it's a metamorphic protein, you will see the intensity change. And I forgot to mention, in the experiment, we actually have a positive control. So this A1 is from John's lab, and they have purified, designed this protein to be metamorphic. And even better, they have a mutation that will lock the protein to not be able to full switching. So you can see the blue line, which is not the one that cannot switch, the intensity will be very similar. The one can switch the conformation, the intensity will change. So by analyze all the protein we can detect from the mass spec. And these are the list of interesting protein I show you. There are others. And today, I will focus on this protein called HINT. You will see what's the reason later. And one of the reasons was also predicted in one of the computational papers in 2025 in Nature Communication, saying this protein is a potential metamorphic protein. And this is another view of the data. Look at all of the peptide that is identified. So you can see this protein, this particular peptide is always it's very strong to be a potential metamorphic protein because this y-axis is basically the slope of the figure I just showed you. So if it's flat, means it's not metamorphic. It is decreased because the temperature change caused the cleavage to change. Then the intensity, the slope will be higher. So you can see the protein I will focus more today. HINT1 HINT, even though it's not very high, but there's a reason we are interested about it. One, it's predicted to be Second, also we have some NMR conformation from the collaborator. It's really likely to be a potential metamorphic protein. But for other new protein, we identify in this screen, again, these we have to validate. And why is NMR the best asset? Because NMR, you can detect the protein conformation present in 2 steps at the same time because it's the solution structural method. Of course, later but the problem for NMR is you only can do small protein. Any protein bigger than 50 kilodalton will be more challenging for NMR. And then we need to develop like cryo-EM method to check the protein conformation or structure at different temperature. Also, I did the functional enrichment analysis of what are the proteins are co-denaturing. So basically are the radar in the graph I just showed you. And they are involved in a lot of fundamental process like translation, peptide biosynthetic process, and regular translation response to heat. So these are, we just want to know what does those proteins do globally. And then these are this table picture to, again, put those proteins in with known information. So this functional enrichment analysis based on what is known in the literature, right? And you can see HINT is over there. I put a red arrow here. It's not linked to anything yet because this is a quite understudied protein, I have to say. So they don't actually link it to any function. And hopefully today I can convince you it's important to regulate protein translation in a specific step. So E. coli HINT is 50% identical to human HINT1. What does this mean? Because it's really important. So evolution keep it, and this protein is named stand for histidine triad nucleotide binding protein. So you can see this region of a lot of histidine. These are the active site where this enzyme bind to the nucleotide, and it conserved between E. coli and human. Okay. Now, this is again just show you that highlight the 3 histidine they are conserved among different species, and these protein actually already have S-ray structure because more than 20 years ago, people already, a little less than 20, shown it interact with protein kinase C and it inhibits protein kinase C, even though later they show it's not. So you can see there's a beautiful active site interact with this AMP structure. And at that time, what is the known substrate for this HINT? It hydrolyzes this phosphoramidate bond. So this is the phosphoramidate hydrolase come from. And I gotta say, in the beginning, they're known as a PKC, and it's a homodimer. And this is how the enzyme works. And through different experiments, we know that this hydrolysis intermediate. So the active site histidine will attack the substrate and will form this AMP HINT intermediate. And water comes in, hydrolyze it, will release the enzyme and the final product. So the reason when I was a PhD student, we studied this because my PI's lab are interested in this prodrug design. So what is a prodrug? It's not active until it's bioactivated inside our body. So everybody knows about COVID, and everybody also knows about the first antiviral drug Gilead developed is a prodrug. And this drug need to be activated by HINT to be functional. So if this particular person has HINT mutation, either loss of function or the mutation in the active site, the drug will not be active because it will stay in this prodrug form. It's not going to be able to inhibit the viral duplication. So my PI's lab at that time, not for COVID, that time we studied we studied HIV, different antiviral, also for cancer. Those prodrug need to enter to the cell. And in the end, these HINT need to hydrolyze those drugs to be able to make the drug active. Then, since then, HINT1 has been implicated in cancer and different brain disease. Even genetic mutation on HINT will cause this inherited peripheral neuropathy. And this is a rare disease, but literally there's already patient secret issues this mutation on HINT cause this disease. And this is just to show what are those amino acid change has been identified in those papers. So you can see, even though this small protein, there are these multiple mutations identified, and some of them has clear link, but most of them essentially are loss of function. Loss of function meaning reduce the enzymatic activity or make the protein not stable, got degraded, or mRNA got degraded. The nonsense mediated decay. Regardless, is loss of function of HINT. So when I mentioned HINT is phosphoramidate, right? So we developed assay at that time to measure how efficient HINT can hydrolyze its potential substrate. So this just show you this is a nice continuous assay. You add the enzyme with the substrate, can measure. You don't need to do multiple time points. And with kinetic, steady state and pre-steady state kinetics, we can prove this. This slope shows you there is the intermediate of enzyme and the substrate can form. So, and these HINT, both E. coli and human, are very efficient enzymes. And the reason why that's important because when we can design a method to trap what is the natural substrate for HINT, because in our body, if you don't go take the drug from here, we don't need to hydrolyze this phosphoramidate, right? Because there's no phosphoramidate there. Why do we need HINT? So the original hypothesis was that on the protein, we all know the protein has lysine group. And if you react with any nucleotide triphosphate, and there's a putative transferase, you might form this protein, either AMP or GMP intermediate, and HINT job might be go there to remove this PTM. So this is basically potentially a one type of post-translational modification, or you need HINT to reverse. So HINT might function like in the kinase phosphatase field, like the phosphatase. We want to remove the PTM. So I spend our time doing this P32 labeling, the old days method, because we can buy the, I should go back here. This is here. So those nucleotide triphosphate, this phosphate can label with alpha P32. The beauty for alpha P32 is very sensitive assay. If anything labeled with the phospho P32, it can read out by a phospho imager. Ideally, these days, we should be able to also do this by mass spec. But in late 20 years ago, this is the best assay to find potential PTM. So this is just one figure I want to show you. I tried it with gamma, alpha GTP, gamma GTP, alpha ATP, gamma ATP. The reason is, at that moment, I don't know which one is the best substrate. So you can see this is the one evidence we can say HINT potentially function as enzyme hydrolyzed phosphoramidate modification on a protein. And this 59 kilodalton is unknown protein, right? Because we only know it labeled by alpha GTP, and the intensity reduced when you have HINT there. So this is without E. coli HINT. You add in purified E. coli HINT, the band disappear. So what I did was I cut this. Well, you need to wait for at least a month. You want the alpha P32 to decay. Otherwise, it's radioactive. Luckily, P32 half-life is short. It's only like 2 weeks. So we wait for a month at a time, and we cut the band from here and to analyze by MODI-12, another mass spec method. And at that time, identify this protein is a potential lysyl tRNA synthetase. And the other experiment I did was, well, I show you the top, right? Because I want to see this 59 kilodalton pen disappear. Here, the same experiment I did also with ATP. Can you see the most striking band on this P32 is this? And I try to explain to you, there is the HINT intermediate. When they hydrolyze the substrate, we will form this potential intermediate. So in the beginning, I don't know what it is. And when I first saw it, I was shocked because this is labeled very clearly, right? And And I hope I can convince you with all this data, and some of this is published before, but now we will validate by mass spec. So what you are seeing that then is basically a potential intermediate in the tRNA synthetase. When the amino acid, let me see if I have, okay, here, ready, static. So when this is the enzyme in our body, and we have 20 different amino acid tRNA synthetase, because each amino acid need to be activated by those tRNA synthetase. And the first step of this reaction is amino acid will bind to the active site of this enzyme, and you need the ATP there because you want to activate the amino acid to make it ready to be transferred to the tRNA. And this is important because it's the building block of all of our protein in our body. So HINT potentially can function as this hydrolase for the amino acid adenylate. So this is the structure of potential amino acid adenylate. So here is just, I did a lot of PCT2 labeling with different conditions and to show you that reaction happen. So the intermediate of the band you see here, basically is HINT hydrolyzed amino acid adenylate near tRNA synthetase. And this is enzymology to show, if we make an analog that is not identical to amino acid adenylate, because if you put the amine group here, that is very unstable. It will hydrolyze by water. So we make this without amino group, and we can hydrolyze with almost close decision limit second order reaction, meaning it's a very potent enzyme, very efficient enzyme, hydrolyze this substrate. And I just explained to you here, essentially, normal situation is the tRNA will come in, right? And the tRNA will be charged with the amino acid. And then we'll go to our body and we'll put the amino acid on the ribosome and to make the protein. So when HINT is not there, what can happen is certain tRNA synthetase might make error in the first step, right? Because, say like tRNA synthetase, that amino acid, arginine, look very similar to lysine. It may not be able to distribute by the tRNA synthetase. So the HINT can function to hydrolyze wrong or incorporate amino acid adenylate in this step. So this will prevent translational error to occur. If you don't prevent it, it means you make the wrong protein, and sometimes the wrong protein might not be misfunction, but when the mutation the substitution occur in the very important protein, then the protein will be misfunctioned or even aggregate or got degraded by the proteasome or protease. And this is just another evidence. People in post-human, which is a big tRNA synthetase guy who sent me the plasmid for LYCU I mentioned earlier. HINT actually can incorporate, accommodate different side chain on the amino acid. In this paper, he used Lysio, AMS, these are the amino acid adenylate analog, or aniline, or threonine, regardless which one, you will bind HINT. So this I just want to show you, HINT can accommodate multiple amino acid adenylate. This, I just want to show you how much time I spent as a PhD student doing all these biochemical assays. But the big question is the top. What are the physiological function of HINT, right? So luckily, when Yanqing, my postdoc, joined the lab about 3 years ago, he was excited about this project. So he's willing to spend his time and investigate what is potential function of HINT. So we have two hypotheses. The first one is what I just showed you, right? The wrong amino acid got incorporated onto tRNA synthetase. HINT's function is to hydrolyze these intermediate here. So if you have HINT here, then the wrong amino acid might be prevented to be incorporated to amino acid tRNA synthetase, this adenylate. If HINT is not there, then the wrong amino acid will be incorporated, so that the mutant protein will be generated. And then in the data I'm going to show you, we think this process happened. So this is kind of the first time we can demonstrate without HINT, the cell will make more single amino acid substitute protein by the mass spec, and also there's a functional consequence of those protein. And luckily, there's a paper from this molecular cell in 2019. They already established the pipeline how to detect single amino acid substitution from the mass spec data. We basically, incorporate the analysis pipeline to find the amino acid substitution. So you can see this is just a big heat map to show you this is the wild type at all. But before that, he needs to make the E. coli knockout. He needs to delete HINT from E. coli. And these are two type of strain. One is the wild type, one is the HINT knockout. You need to run 3 duplicate mass spec, and to go through this pipeline that is established by the paper I just showed you, the molecular cell paper, and to identify this so-called mutation frequency. So you can see some amino acid, you can clearly see in HINT, it has more mutation. But like I said, translational error is actually one of the area, especially in the so-called pre-transfer editing. Before the amino acid got transferred to the tRNA step, it's called pre-transfer editing. In this step, the translational error is hard to measure, right? Because genetic error, you can sequence the DNA sequence. But amino acid mutation, people studied that with the reporter until recently, and this is a bioarchive paper from Michael Lin. Mass spec is the only method. I just highlight red here. Mass spec is the only method at this moment you can detect single amino acid substitution at scope-1 scale. Because if you don't measure the amino acid sequence or the peptide sequence, how do you know the real mutation rate in the species? But we need to validate, right? The heat map I just show you some amino acids tend to be misincorporated when HINT is knocked out. So EMP need to go look through the data to find the peptide. And this is show you he can validate in a cell-free system. Cell-free system, basically you can buy a kit from AEB. They contain all the transcription translational machinery. And you can add HINT without adding HINT, see which amino acid, which peptide, the incorporation of that single amino acid change is decreased when HINT is added. So without HINT, the error is more. When you add HINT, the error decreases. So this is, and these are, he picked a few peptides from the proteomics analysis. Now, that's not enough to show, how HINT might regulate tRNA synthase for the translational error. Then he did a next experiment, it's called thermal proteome profiling. And again, this method has been developed by other people to look at, say, protein and drug interaction. They use this method to identify what is the off-target of this potential drug is being used. And we also do that a lot. But there's another paper, and so the drug interaction paper is this 2018 paper. Then later, there's a lab, they knock out a bunch of different protein in E. coli. They didn't do HINT in their paper, but they knock out a bunch of protein in E. coli and do this thermal proteomic profiling. And what is a thermal proteomic profiling? So I think that, here, this is easy to describe. They want property. So basically, each protein has its natural thermal melting curve. So which means like when we are cooking egg, right? If you increase the temperature, most of the protein will also denature or become solid. And each protein, when you increase the temperature, and 50% of the protein that denature, that's called melting temperature. So in all that biochemically, we measure one at a time. We can run an SBS test, we can run a Western blot. So essentially, the 2018 science paper that use this method to proteome-wide to get as many protein as possible in the sample they can prepare with or without drug. So then anything has the melting temperature shift is the potential interactor of the drug. In the Nature 2020 paper, they compare wild type E. coli and a protein knockout, a gene knockout strand, doing the same experiment. If the protein they form a complex, they potentially will melt together. So if the knockout affecting a melting temperature of the other protein, is an indication of these two proteins interact. The same thing as a drug. So if the drug binds to a protein, the thermal stability change is a way to suggest this drug and this protein interact. The caveat is if nothing changed, it doesn't necessarily mean they don't interact. So only the positive result tell you something. So Yanping used this method. He did the TPP, thermal proteome profiling, for wild-type E. coli and HINT knockout E. coli. So then he can identify a potential interacting protein. Surprise! So we find a lot of tRNA synthase in this protein-protein interaction network analysis, and also some like ribosomal protein. So we are excited to have another, I will call unbiased proteome-wide scale analysis, because if you go purify one protein to measure the interaction, you can always say, oh, maybe just pick that one. So this, at least, I do believe more, it's more global. We try to look at as many proteins as possible. I didn't show you other data. He also purified this protein, those tRNA synthase, and measure one at a time to make sure they interact. So now, E. coli is a good way to start to do proteomic study. But E. coli doesn't have a lot of good way to check the phenotype, right? And for the mouse, if you knock out HINT, there's some conflicting data. So there's one paper saying there's no phenotype. There's another earlier paper saying you knock out HINT in mice because it's a heteroinsufficient tumor suppressor. So it's a tumor suppressor, but it's not 100% sufficient. So, and like I told all other postdocs, my lab don't work with mouse. So we collaborate with, my husband, who is still the lab in his lab post number. So Yanping from my lab and Yinan from post lab, who create HINT knockout in C. elegans. And we try to observe phenotype. We also try to do the translational error measurement by mass spec. So you can see this assay is widely used in C. elegans field to look at the phenotype. And you can see that when you knock out HINT, the number of progeny decreased a lot, significantly. And what Yanping and Yinan even do more, if you express the HINT, you can rescue, and you can rescue by both E. coli and human and C. elegans HINT. And these are just a different way to view the data. And Yanping even goes a step further. He wants to understand what happened during this embryogenesis. Why does those eggs don't hatch? So there's no progeny from C. elegans. Actually, there are some, not 100%. It's not 100% penetrant. Not all the eggs die, but like there may be a percentage survive. And he want to figure out, which is hard. I don't think we figure out yet, but at least what he did is pick up the embryo at different stage from C. elegans egg, and you can see compare wild type, this side E, and the HINT knockout. You can see they are really weird. It's disformed. The structure of this development is different, right? So we know they die at certain stage of embryogenesis. But again, why? And we, of course, believe it's because HINT might be important during, or the protein translation load might be higher during this earlier stage. So when you don't have HINT, then you tend to make more mutant protein or single amino acid substitute protein in your stage. This is my model. Yeah. So, I think we already 45 minutes. If there's a question for this section, you should ask me. There's another protein. I don't want to give you too much data until we understand what HINT is doing first. And there are other data I didn't show you, like for this E. coli, for the C. elegans knockout, we did top with ubiquitin conjugate, and we also have human HINT knockout in the HeLa cell. You see a lot of ubiquitin conjugate. What does this mean? There are misfolded protein occurred when you knock out HINT. And the limitation of this study, in my opinion, at this moment is we want to go figure out why and how HINT can recognize incorporate amino acid adenylate in the active site, because when you do biochemically, now the substrate specificity is quite similar, because HINT has its open active site, can bind to different amino acid adenylate. So there must be a reason or a factor that tell HINT, now this is the wrong amino acid, you need to go hydrolyze it, otherwise this wrong amino acid will be incorporated into protein, and it's not good. And we also believe potentially these are also why there are so many different diseases. HINT is involved, and there's not a central thing of what is the particular pathway, because maybe in one disease the protein translation burden is higher than the other. Therefore, it's not just one particular pathway at this moment associated disease, but it's involved in multiple diseases at this moment. Okay. Thank you,
Dr. Chou. So yes, now we have time for questions.
Yeah, thank you so much for the really fascinating HINT protein. I never heard know about that before this. I have a quick question. Does all the different cell types have the HINT protein, or?
Yeah, so at this moment, it's really widespread, and yeah, but HINT has 3 flavors. There's HINT1, HINT2, HINT3. So for HINT3, I think there's more cell type specific. HINT2 is in mitochondria. So there's a mitochondria targeting sequence for HINT2 to go to mitochondria. But that's also for future, right? We want to know why you need a specific HINT. And it's about 80% sequence identity to human HINT1. The main difference is the mitochondria targeting sequence. Mitochondria don't make a lot of protein, right? As the protein comes from imported. So why do you need a specific HINT there? But HINT1 itself, the conserved one, like served to E. coli, that one is everywhere. It's everywhere.
Interesting. I'm just wondering if any of the dysfunction of the HINT protein, does it have any studies yet to any neurological disease?
So the disease I show you is called peripheral neuropathy. Yeah, but for the brain, there are there's opiate addiction leading to think also. Yeah.
Any other questions?
Thank you.
Yeah.
Thanks for the nice talk. As a HINT, it regulates the blood size. Does it also have any effect of schizophrenia disease, like brain disorder or some other?
I think I gave you that quickly for the disease relevant. Yeah. So those are a lot of mood disorders or with the schizophrenia, anxiety, they all linked. And the opiate addiction or something, there are a lot of these papers, but the mechanism is not known, right? So I do think, yeah. So if there's any interesting or hint in the disease here, happy to collaborate.
Yeah, thank you so much. And is there any roles of HINT in the neurodevelopmental disorder and in the context of neurovascular aspect?
Now I know of at this moment. So, but not from the literature, but I don't know if people have looked specifically, or if there's a genetic mutation people identify. I don't think so.
Thank you.
Thank you for a great presentation. For pain and analgesia, do you think HINT expression changes for people who have pain and no pain? And is there post-translational modification of HINT when the levels change?
In the pain study?
Yeah.
In the pain analgesia, I know my PI Wagner, they try to develop actually inhibitor of HINT. So the prediction would be you want to inhibit its function because its function there cause change.
Oh.
Yeah. Wait. Or as analgesia, I can get back to you on that one more specifically. But the inhibitor is what they try to develop. Yeah.
That was different from what I was thinking. I was thinking that the inhibitor will prevent pain.
Let's talk later. I focus more on the mechanism. No, I just did here. Yeah.
Thank you so much.
No, they took an opiate. So let's deal with HINT later. Pain later.
Yeah, I have a question regarding the technique, which is the thermal proteome profiling. So, in terms of detection, how the concentration, how low it can detect? I have three questions. One is what is the lower limit of detection for this technique? And the second question is how many protein-protein interactions for a targeted protein of interest it can detect? And the third question is what are the limitations of this technique in terms of while we are looking into protein-protein interaction?
Okay. So for the first question is for the thermoproteomic profiling, you do need to have if you look at proteome-wide scale, right? If you say you have one type of cell and you add a drug, then we typically use cell line at this moment. People can also get from the mouse, but there's a in vivo TPP. And so it's the initial material is a lot because we need to split them in different temperature. So I would say 5 milligram to 10 milligram of the starting material of the total protein you can isolate. And if you have a specific protein of interest, then it depends on abundance of that protein, right? But again, the mass spec is more sensitive these days. If you only care about your protein, then the method can adjust more specific or more sensitive. But typically, when we do unbiased, we want to detect as many protein as possible, right? So in that case, we just, you input as much as possible. But when you know on the mass spec, you don't know all of it. You can still keep it. So for the cell line or lysate, we normally start with 5 to 10 milligram. And so that's the amount you need. Second question is how many protein you can detect? Essentially, like the limitation detection for in one cell line, 5 to 10,000 protein depends on the cell line. The third question is the limitation, right? I try to mention that because not all the interaction will cause thermal stability to change. So the limitation comes if this protein-protein interaction does not induce thermal stability to change, then you cannot detect. So the negative result does not tell you they don't interact. Only the one shift can tell you they potentially interact. Does that answer your question?
Thank you, Dr. Chou, for this wealth of information. Can you comment on the size of these metamorphic proteins? Are they always small? And I was wondering, since they are shape-shifting, they're probably evolutionarily conserved, right? And it's probably to do multiple functions in different times.
Yeah. So I think the reason at this moment why most of them are small, like I mentioned, the technique to detect it to observe it two-step is by Engemann. Or there's another method, it's called site exclusion. If you have you can separate the protein complex by different sites. Similar to TPP, you can have the whole lysate and you separate by different sites, and you detect each protein complex in different fractions. And if you do find some protein exists in multiple fractions with different partners, you can then go study other potential metamorphic protein. But for the technique to observe it, simultaneously happen with Engemann technique. So then Engemann technique limited by protein size. And there is also another method, it's called hydrogen deuterium exchange. It's by mass spec, but you can only, like Engemann, you can only do one protein at a time. So you kind of need to have some evidence to point to you, this might be a potential metamorphic protein for you to do those experiments, since we cannot predict by sequence. There are some computation methods try to predict, but again, they are not perfect yet. So the hope is with our proteomics method, we can generate a potential list. And then we will start with the low-hanging fruit. We will do the low molecular weight by Engemann, collaborator Andy and John will do it. And then we can validate they are metamorphic protein. And the next step we can do is, why do they need to switch shape, right? like you mentioned, it might change different function or change the protein-protein interaction. So we can then design the mutation to last at the particular step, or we can just do cool down at different temperature to identify the protein-protein interaction network change of this protein. And actually, we already did that for HINT protein, and we did see at lower temperature and higher temperature, the protein-protein interaction pattern does change. And I do also agree with the evolution concept one, but then the problem is that we need to find them first to really analyze them to see how evolutionary concept. And there are some protein like cell cycle regulator protein, MAD. So there's this domain structure people already know is a potential region for switch. And they can use that particular region to identify the other one. Like the other protein I didn't talk about, ATG101 today, we did not identify it's a metamorphic protein. Other people already identify that one is a metamorphic protein. You know what I'm saying? But through the region of there's a concept domain in the other protein, and they already think it's metamorphic. So then they do the exact exclusion method I mentioned to show, wow, there are different complex can form, and their interaction affinity this change. So show that they are potential metamorphic.
Thank you.
Any
or question just coming out. The first thing is when you testing the human samples, does the HINT protein have sex difference, females and males?
And we did not test the human sample yet. We take human cell line
Oh, okay.
data. Yeah. So yeah, I don't know. We can check with the database. Why do you think there might be a potential sex difference?
Yeah, because you just mentioned about the evolutionary role.
Oh, okay.
So the female and the male probably play different side of reaction, different responses.
Okay. We can check. I'm not sure.
Okay.
Yeah. So the human in my lab, it's more human cell line or iPS derived cell. That's the most human we do. There are other human sample projects that not on HINT collaborate with other people.
Yeah, the second question is for the human, the neurotransmitters, because we look at the brain waves, we roughly separate them to excitatory and inhibitory. So the HINT protein, are they more interactive with the excitatory or inhibitory?
We don't know. I don't know if people know.
Oh, okay.
Yeah, we can check and look. Yeah, see if there's anything interesting there.
Thank you.
Thank you very much for the presentation. The HINT1 deficient mice develop tumor instantly when they get old.
Mm-hmm.
And my question is, the second one is whether this neurological disorder happen at an advanced age as well, or they happen at birth.
Oh, I think they are not at birth. The one I know inherit is it's not early onset, but for other disease like Down syndrome, the earlier, right?
Yeah,
the mouse model study. So there is the earlier study, age related and they have symptoms. But Jackson Lab, they put another paper out, more not recent, right? More newer than the previous one. They can prove knockout HINT and they in their hand don't have strong phenotype or so we actually want to figure out if there's the other collaborator want to redo the mouse model, or we can just buy the model from Jackson we kid experiment.
Yeah, the question is, because they have the knockout, they have an issue with tumors and brain disorder. And these are two different parts. One is tumors, so would involve more the immune system, while the brain, I would involve more neurons or endothelial cells or even microglia.
Yeah.
And my question is whether you looked in the context of aging in HINT1 expression or change.
And not yet. We haven't looked at this yet. Yeah, but other people know.
Yeah.
So for me, the exciting part, which we think we I just coming one postdoc 3 years ago, is really, I want to understand what is the fundamental physiological function of HINT, right? If we don't, then to think the disease is trickier. And if we really believe it's the translation error, then we really need to also go look every phenotype we see in those genetic disease. I want to see if the translation error occur. Now we have a pipeline we can do. There's another one, the PTM, the post-translational modification. I still believe it's possible doing that rule. It's just the tool to study there is slightly harder because we have to, those modifications, the phosphoramidate bond I show you, those are acid labile. So you cannot do a normal LC-MS profile we normally do because we put in a low pH. So we have to optimize the method, change the LC condition, and to dedicate deep dive to see what are the potential PTM things might also regulate. So these both are really broad, right? So it's the translational or the PTM affecting the PTM function in those disease proteins would be the important next step. And a lot of correlation data, like genetics one, I highlight that genetic because literally this disease has genetic mutation. The other one is all correlation observation, and yeah, we need to really do a normal to really know in other disease how do we treat the patient if HINT is a potential way to help. Yeah.
Thank you so much. Do we have one more question?
Yeah, any last questions? I don't think so.
Thank you. Very exciting talk.
Yeah.